Hi, my name is Niela Vibreska, and I will be presenting our poster for our Enzyme Lab. Enzymes are proteins which are in charge of speeding up chemical reactions, making them biological catalysts. They do this by lowering the activation energy of reactions. They are extremely important in living organisms because most biochemical reactions are so slow that they wouldn't occur under the normal conditions of life. Enzymes allow for the regulation of these reaction rates, so they speed them up. Trypsin is the enzyme that we were focusing on, catalyzes the hydrolysis of peptide bonds. So it breaks them down, breaks proteins down into smaller peptides. It cleaves at the C terminal, which is the carboxyl side of the amino acids, either lysine or arginine. Trypsin's role in organisms is breaking down dietary protein in the small intestine, where the pH is approximately 8. It is first secreted by the pancreas, where pH is variable, and it's secreted inactively, so it's in the form of trypsinogen. So our question for our project was if temperature has an effect on the activity of trypsin, and our hypothesis was that at 60 degrees Celsius, bacterial trypsin will be at its optimal activity, and any temperature below or above this will denature the enzyme, which will slow down the enzymatic activity. The reason why we made this hypothesis is because we researched at which temperature bacterial trypsin functioned best, and we found that it was at approximately 60 degrees Celsius. And we did that after realizing that we predicted the optimal temperature for trypsin in humans, which was 35 degrees Celsius for our first experiment, rather than the optimal temperature for bacterial trypsin. The materials that we used were 0.5 molar sodium chloride with a pH of 8, and that was our reaction buffer, azocasein, which was the substrate that was broken down by trypsin, and trypsin, our enzyme, 10% TCA solution, and temperature-controlled water baths at which we tested different temperatures. First, we added 450 microliters of our reaction buffer into the centrifuge tube, followed by 500 microliters of azocasein. We then added 50 microliters of our enzyme trypsin, and we incubated the tubes at 0 degrees, 23, 30, 45, 60, 75, and 90 degrees Celsius for 10 minutes. The tubes were then removed, and TCA was added to stop the reaction and precipitate the undigested substrate. Then they were centrifuged at a maximum speed for 2 minutes to remove the undigested substrate. The supernatant was transferred into a clean cuvette so the absorbance could be read by the spectrophotometer at 440 nanometers. The addition of sulfanilamide is correlated to the measurement of absorbance at 440 nanometers because azocasein is a milk protein with sulfanilamide attached randomly, so the sulfanilamide allows it to absorb at 440 nanometers. Our controls for our experiment were the reaction buffer. We had a salt concentration of 0.5 molars and a pH of 8 to mimic the conditions at which trypsin is active in the small intestine. For our negative controls, we did not add trypsin at the temperatures 0, 45, and 90 degrees Celsius. So we expected no response from these negative controls, and it is helpful to add negative controls in experiments to see if we made any mistakes with our positive controls where the trypsin was added. And we also used 3 replicates for each temperature, and we used 3 replicates for each negative control. The use of these replicates strengthens the conclusions that we draw from our data because we can see if there are any outliers and eliminate them when we are analyzing and interpreting our results, and we can take them into account when we're doing our statistical tests. So these are our results. So we showed them here in this bar graph, and you can see that it has a normal distribution, so the absorbances are lower for 60 degrees Celsius and lower after, so that's what we wanted to see. We also see that the average absorbance was highest at 60 degrees Celsius, which is also what we wanted to see. But we can't really conclude anything before we go and do our t-test. So our t-test is also on our side because it tells us that these temperatures, each temperature is significantly different than 60 degrees Celsius. So that we can conclude that there's a significant difference between the temperatures. And our negative controls, we can also see that they're different from our positive controls, which is why we use them to reference back on. So we could, for example, a control average for our negative controls was 0.018 at 0 degrees, and the positive control was 0.058. So there's a difference because no trypsin was added. So the standard deviations were also relatively small. The largest standard deviation was 0.019, and that is good. So we saw even in our results there's not a lot of variability, which is also a good sign, and it's helpful to have replicates so we could see that there's no variability. So in the end, this experimental design was adequate to test our hypothesis. Our results supported our hypothesis because our t-tests show that 60 degrees Celsius is significantly different than any other of our temperatures. All of our p-values are under 0.05, which is what our t-tests were testing against. So other groups, they also had a kind of normal distribution of their data, rounded like distributed lower and higher from lower on both sides from their prediction for the optimum temperature. One thing that we could do to really find out what exactly is the optimum temperature for trypsin is test more temperatures between 30 and 75, like a wider range, because we know that it's higher at 60 than all of these, but we don't know if it's exactly 60. So that's a change that we could do in the future. Some sources of variability that we could have had were a couple sources of variability that could have happened without us realizing. Could have been not switching the micropipettes between like taking out the absorbances, like when testing the absorbances using the spectrophotometer, not changing the micropipettes. Another source of variability could have been not cleaning the cuvettes with the clean wipes when taking the absorbances using the spectrophotometer. This experiment was a lot more successful than our first experiment because our first experiment we tested that the optimal temperature would be 35, and we didn't get this even distribution of our results. So for future attempts, as I said, the only thing that we could do to make this design better would be tests. Now that we know that 60 degrees is optimum, is significantly different than these temperatures, then we could test between these temperatures to see exactly where trypsin is at its optimum. In conclusion, this shows us at which temperature trypsin functions best. So if we were to grow bacteria, it would be in an environment around 60 degrees Celsius, and that would have shown the